So the next step of the molecular cloning is the ligation of the vector and the insert DNA. So in the previous step, we have prepared the DNA and the vector either by digestions with a restriction enzyme or by PCR. But now we want to make these two fragments join together and make a one DNA fragment. And this is called a ligation. So in this step, both the vector and the DNA fragments are taken in a one single tube and mix them with the DNA ligase. That is the enzyme that facilitates the ligation of the vector and DNA. So we need to keep in mind that when we do the restriction digestions, we need to use the same restriction enzymes for both the vector and the DNA so that they can produce the similar or same sticky ends and then join together. So the DNA ligase normally facilitate this ligation of the vector and the DNA in a three-step process. At the first step, the ligase reacted with the ATP, that's the adenosine triphosphate, and form a ligase AMP intermediate. Ligase AMP intermediate. And then in the next step, this ligase AMP intermediate transfer the adenosine monophosphate, that is the AMP, to the 5' phosphate group, which has been cleaved during the restriction digestions. That's the unprotected phosphate group. So the AMP is joined to the phosphate group of that cleaved sequence. And then at the next step, the hydroxyl of the 3' group of that cleaved side, this is 3' end of that cleaved side, the hydroxyl group is here. So this hydroxyl group then interact with this 5' AMP phosphate group and sealed the joint together. So this is how the ligase joining the vector and the insert DNA. So after ligating the vector and the DNA, so we have in a centrifuge tube the ligation mixture, and we don't know actually whether this vector and the DNA are ligated or not. So what we need to do is transform this ligation mixture to the host organism. In most cases, this is the bacteria, that's the E. coli bacteria. And if you can recall that we need to prepare or select a host organism, mostly the bacteria, and make it competent so that the bacterial cell wall is highly porous. I mean, just make it a porous so that the DNA can get inside of the cell. And you need to also can be remembered that there are two processes we need to make the competent cells. One is the chemical method that is mostly using the calcium chloride, and the other is the electroporation that is using the electricity. So in the chemical method, which is mostly used into the laboratory, or you can also find this one into the biotech industries as well. So in this way, so when we do the chemical transformation, we need to use the competent cells which are prepared by chemical method. So in this state, the competent cells is treated with the calcium, as you have said, and create a pores to the bacterial membrane. So this also, the treatment also may or also mask the negative charge of the DNA and promote the binding of the DNA to the membrane. So when there is a heat shock step, there is a heat shock at 42 degree for, I think it's 90 minutes, 90 seconds. So when this heat shock is given, there is a thermal gradient generated, and that thermal gradient is facilitate the DNA to move into the cell or the bacterial cell. So this is a chemical transformation. On the other hand, we can also use the electroporation or electrically competent cells when we have the electrically competent cells has a higher efficiency than the chemically competent cells. So we can use this electroporation, but we need to have the electric equipments to do this process. So in the electroporation transformation, the electric current generates pores on the membrane of the cell and where the DNA is actually trapped. So as soon as the electric supply is removed, those pores are closed and DNA moved inside the cell. So these are the two main transformation procedure generally applied into the laboratory to transform the ligation mixture or the DNA into the competent bacterial cell. So what is happened after the transformation to the bacterial cell? So the cells were then plated out into the agarose media with appropriate antibiotic into the media, as we expect that our vector, in most cases, having an antibiotic resistance gene. So those are the bacteria will grow into that plate. Those have this antibiotic resistance gene, or those bacteria which are actually taken up those vectors that is having that antibiotic resistance gene. In most cases, ampicillin has been used widely, ampicillin, for selection of this bacteria. In other antibiotics like carbapenems or chloramphenicol, tetracyclines, or kanamycin has also been used. So this is called the antibiotic resistance. This is just the first step of that. Yes, we got a transformant, and we know the bacteria is taken up the plasmid or the vector because it has the specific antibiotic resistance gene. Then we can also another screening procedure like a blue and white screening, which we will discuss a bit in the next slide. And also we need to confirm this with a colony PCR and a colony hybridization. So we will discuss these things in later. So in the agar plate, any bacteria that has taken up the vector, which has the specific antibiotic resistance gene, will grow. But that does not mean that the bacteria is, not the bacteria, that the vector is actually ligating with our insert DNA into that plasmid. So for this, there is a one visual screening, which is a blue and white screening. So what happened in this procedure is that the bacteria has a lac operon. So this is mostly the metabolization for lactose or its related product. So just producing the enzymes that actually metabolize the lactose or its related product like IPTG. So in this lac operon, there is a gene, this called LAC-Z. And this LAC-Z gene is actually encodes an enzyme called beta-galactosides. And this beta-galactosides, actually production of this beta-galactoside is induced by the lactose or IPTG, as I said before. So when expressed, the beta-galactoside enzyme can break down a dye or dye-like substance called XGAL into galactose and a blue pigment, insoluble blue pigment. So scientists, what they did, they actually generate a mutant LAC-Z gene, and it's inserted into the multiple cloning site of a vector. So what happens when the insert DNA is ligated to the multiple cloning site of the vector? The LAGZ gene is not expressed. And when it is not expressed, it cannot produce that beta galactosidase enzyme. And the enzyme cannot break down the XGAL substance, the substrate XGAL. And as a result, we got white colonies. On the other hand, when the insert DNA is not ligated into the multiple cloning sites of the vector, that is, the vector is actually religated. And we know that this is possible. The vector can be religated by itself. So that in that case, the LAGZ gene is actually expressed and it produces the beta galactosidase enzyme. And as a result, the enzyme then break down the substrate XGAL and produce that blue colors, or in the plate, on the plate, we can see there's the blue colonies. So this is a one type of visual screening that gives us an idea that after the transformation, how many colonies we get with an insert ligated to the vector, or how many colonies are just self-ligating by itself. So as I'm just explaining the blue and white screening method, so there are like a three scenario will be coming up. Number one, the vector is not taking up the insert and they are religated, or self-ligation happened to the vector, or the vector is ligated with the insert, which is the expected one, which we want, and the insert ligated with each other, which is very rarely, but it could be a possibility. So in the first case, when the vector is actually ligated by itself, it will still grow into the plate because the vector has the antibiotic resistance gene. So it will grow, but it will grow as a blue colonies because the LAGZ gene has been expressed. The DNA insert is not ligated into the multiple cloning site. So the LAGZ gene is expressed, produce the beta galactosidase enzyme, which break down the substrate XGAL and producing blue colonies. In the second case scenario, when the DNA insert is ligated with the vector, that time the LAGZ gene is not expressed, no beta galactosidase on the plate, and there is no blue colonies. We got some white colonies, which we want for our experiment. And in the third case, if the insert ligate to each other, in that case, we should not get any colonies into the plate because there is no vector at all. And if there is no vector, the insert doesn't have the antibiotic resistance gene. So if the insert ligated between each other and still they go into the bacterial cell, but the bacteria can't grow because the plate or the media has the antibiotic, and for growing of the bacteria into that antibiotic plate or antibiotic media containing plate, they need that specific antibiotic resistance gene. So this is the basics of the blue and white screening. This is also called alpha complementation. So I'd just like to request that you can check the video I have attached here, or there are multiple, there are hundreds of videos into the YouTubes, and just go through with some of them. That will make things a bit clear for all you guys.